First of all, I would like to say thank you everybody for joining us today. This is the Daniel O. Trainer Wildlife Health Seminar Series. Today we are featuring Drs. James Peirce and Greg Sandland from UW-La Crosse. And so, first of all, I would like to say thank you to not only everyone for being here, but I'd like to thank everybody who has helped to organize this. My name is Jennifer Summers, and I am the Program Development Specialist with the Wisconsin Center for Wildlife, and I have helped organize this seminar series. But I would also like to thank Shelley Dubé, who was kind of helped pull this idea together, help me think of people to contact to participate in the series. Also like to thank Marie Perkins, and then Ben Settinger, who's our waterfowl specialist, and again, UW Extension, and also the College of Natural Resources for all of their help in pulling this together. So we have a total of 5 presentations or seminars in this seminar series. This is our second, and if you would like to get more information about other speakers in our series here, please check out our website at uwsp.edu/WCW, and there will be a link there for our seminar series. And we have other seminar series that will be happening in the future as well. So check our website often. So we have a agreement with the tribes and Stevens Point, and we do acknowledge the tribes every time we have any kind of a public gathering, and virtual presentations count as public gatherings. So we recognize the University of Wisconsin-Stevens Point occupies lands of the Ho-Chunk and Menominee people. Please take a moment to acknowledge and honor the ancestral Ho-Chunk and Menominee land and the sacred land of all Indigenous peoples. And now I'm going to pass it over to Dr. Dubé, or Shelley Dubé, who's going to say a few words about the namesake of our seminar series here, Dr. Daniel O. Trainer.
Yeah, it's really hard to beat what the dean had to say last week. However, I'll do my best. I'm going to kind of put a little personal spin on it, just because I was a graduate student in the 1990s in wildlife health. And I kept coming across the name Dan Trainer. You know, I was always reading articles on deer and waterfowl and all sorts of things. And I was like, wow, that guy seems like he's pretty important. And lo and behold, I get a job and I get to walk into the Daniel O. Trainer College of Natural Resources building every day and see his picture. And it's just a real honor to be here, because he was brilliant, of course, in wildlife health, but also a really good human being and an extremely funny person. So when I met him once before he passed away, and when people get to talking about him, they would say, oh, do you remember the time when he used to leave like a dead skunk in so-and-so's office and hide it? And then the next thing, hey, do you smell that? He just was a card. It sounds like he just was a man of the students. He was always interested in figuring out the best way to deliver the material to students. And then he also mentored so many faculty members, including our former dean, Dean Thomas. So I'm really happy that we named this seminar series after Dr. Trainer. And we've got some great speakers lined up. So I'm again, really feeling very fortunate. And with that, Dr. Settinger will introduce our speakers.
All right. Hi, everybody. I'm Ben Settinger. I'm in the Wildlife Department. And so it started when Dr. James Peirce complimented Dr. Greg Sandland's Rush t-shirt in the hallway of the science building at UW-La Crosse, where Dr. Peirce is a professor of mathematics and statistics, and Dr. Sandland is a professor of biology. They quickly learned they had many shared interests and have been collaborating together for over 10 years. Dr. Peirce earned his Ph.D. in mathematics at UC Davis, and Dr. Sandland earned his Ph.D. at Purdue. They both have research interests in host-parasite interactions, disease ecology, and mathematical biology. They share a particular pride in the accomplishments they've made co-mentoring their undergraduate researchers. Through their work studying host-parasite interactions and disease ecology, they've worked hard to impart a love of ecology questions and a mixed approach of integrating empirical perspective with mathematical structure. Their latest work involves investigating the role of common mergansers in the transmission of swimmer's itch, which is an emerging parasitic disease. So this should be great today. Everyone, please welcome Dr. James Peirce and Dr. Greg Sandland.
Thank you very much, Ben. I'm now going to try to share my screen here. Is everybody seeing it?
Good.
Excellent. All right, well, thank you so much for having us here today. James and I have been working on swimmer's itch related issues now for 5 to 7 years, and we always look forward to the opportunity of potentially chatting to new groups of individuals about our work. So before I actually get into the development of the mathematical model itself, I want to talk to you a little bit about swimmer's itch and what sort of led us to focusing in on this kind of particular problem throughout the Midwest and actually throughout North America. So swimmer's itch is actually caused by flatworm parasites that use 2 different hosts as part of their life cycles. They use birds and sometimes small mammals as what we call definitive hosts in their life cycles. And then they end up utilizing snails as intermediate hosts in their life cycles. And the problem for us as humans is when we end up coming in contact with these larval forms of the parasite that end up being shed from these snails. And so this is, as I mentioned before, this is a problem that we find throughout the Midwest and actually throughout North America. Some local areas where this problem has emerged, Forest Lake, Minnesota. So a couple of years ago, there was a major outbreak of swimmer's itch at Forest Lake. And for many of you, you know Devil's Lake is a very important recreational water body here in Wisconsin. And 2 years ago, and I think about 5 or 6 years ago, there were major outbreaks of swimmer's itch at that particular location, along with a number of other freshwater bodies throughout both Wisconsin, Minnesota, and through other areas of the Midwest, including Michigan. So when you end up having outbreaks of swimmer's itch, this generates some major problems. And a lot of these problems arise via costs. So a big one is a loss in tourism, because nobody wants to go to these freshwater bodies. Nobody wants to go into the water of these lakes and potentially come down with this particular problem, potentially come in contact with these larval forms of the parasite. And if people aren't going to the lakes, then you can have a loss in tourism and a loss in tourism dollars to the towns and cities that are actually surrounding these particular lakes. So that's a big one. Secondly, there can be healthcare costs associated with swimmer's itch. So some individuals that end up coming down with this problem end up having to go to the hospital and need to be treated for rather severe reactions to these larval worms that are potentially trying to infect them. So there can be healthcare costs associated with swimmer's itch outbreaks. And then of course, there's the cost of mitigating these problems. So getting individuals into these environments, potentially trying to manipulate these environments to reduce or eliminate the probability of swimmer's itch in areas where individuals are actually in the water, swimming, bathing, kayaking, etc. So because of the fact that there are large costs associated with swimmer's itch, and because of the fact that we end up having such a widespread occurrence of swimmer's itch throughout the Midwest and across the globe, swimmer's itch has now recently been recognized as a very important emerging disease. So let's talk a little bit about swimmer's itch itself and the life cycle of this particular parasite. So as I mentioned before, the parasite utilizes 2 hosts to complete its life cycle. Here in the Midwest, one of the key hosts in terms of these definitive hosts ends up being the common merganser. So adult worms actually live inside of these mergansers, and adult females end up actually releasing eggs that are shed into the water column. And usually immediately after these eggs get into the water, they hatch, and they hatch into a ciliated larval form known as a miracidium. And the sole purpose of the miracidium is to seek out, penetrate, and establish in snail first intermediate hosts. And the type of snail that's infected really depends on the kind of schistosome that's occurring in the surrounding water column. So once the parasites get into these snails, after a period of about 4 to 6 weeks, depending on water temperature, the parasite undergoes this really complicated transformation and moves from this myricidal stage to the second free living larval form stage called the cercaria, which are shed from these snails in large amounts. And once these cercaria are shed into the water column, their sole purpose is to seek out, find, and penetrate into these mergansers. And they'll do this through the feet, they'll do this through the legs. Now, the problem for us is that it turns out that a lot of the chemical cues that are being released from the legs of common mergansers are also cues that are released from our skin. And so these parasites will oftentimes try to infect us as opposed to going into these particular birds. And this, again, there are some pros and some cons of interactions with this parasite at this particular point in the life cycle. And the positive is that for us, our immune system does exactly what it's supposed to do. It ends up killing these parasites off before they're able to establish within our bodies. And so we, in the life cycle, are actually considered dead-end hosts. The parasites can penetrate into our skin, but our immune systems kill them off before they're actually able to reach adulthood. So we do not contribute to the cycling of this particular parasite in these different environments. So that's the good news. The parasites aren't actually establishing within us. The bad news is that at every point on our bodies where those parasites try to penetrate into our skin, we end up having localized inflammatory reactions. And you can see some of those reactions over on the right-hand side of the screen. This is a relatively mild case of swimmer's itch up at the top. That is actually my leg. So that is a mild case of swimmer's itch that I ended up getting when I was kayaking Lake Superior a couple of years ago. And each one of these raised papules that you see here is a point on my leg where those cercaria tried to enter my body. This is a much more extreme case. This occurred in a colleague of ours who actually was in the water for a very short period of time, had her legs in the water before she was going into her kayak, had her legs in the water as she was getting out of her kayak, and you can see she must have put her legs into a large cercarial cloud because at points along her leg, it almost looks like a complete rash. But if you look a little lower on the leg, again, you can see each one of those individual points representing where those cercaria tried to enter into her leg. And then finally down to the bottom, just to remind you that this doesn't only occur on the legs or the arms. You can actually have swimmer's itch and these papules occurring anywhere on the body that those parasites have tried to enter our skin. And this can include the upper areas of the body as well, if we, for example, swim through areas where those larval parasite forms exist. Now, people have oftentimes equated these raised papules to something like a mosquito bite. And I would argue that they're nothing like a mosquito bite. They may look like a mosquito bite, but the itch itself is far greater and it persists for a far longer period of time. But in the grand scheme of things, this isn't so bad relative to the reactions that some people actually exhibit. So some people will adversely respond to cercaria trying to enter into their bodies. And this can result in fever, swollen lymph nodes, in some cases diarrhea, lethargy, and in odd instances, individuals have actually experienced anaphylactic shock because they've received such a large dose of cercaria when they've gone swimming, when they've gone wading in some of these water bodies, which can be, again, dire, life-threatening in some cases. So given the fact that this is such a major problem in the Midwest and such a major problem across the globe, what are our options in terms of dealing with this potential issue? And when you're dealing with a complex life cycle parasite, things become a little more complicated. But I would actually argue dealing with a complex life cycle parasite also provides you with more options in terms of potential targets in the life cycle. So if we go back to our life cycle of the swimmer's itch parasite, these avian schistosomes, we've got our definitive hosts, we've got our intermediate host snails, and then of course we've got us serving as dead-end hosts. So what are some of the strategies that we can employ to actually reduce outbreaks of swimmer's itch? And of course, one of the most straightforward ways to do this is to urge individuals to stay out of the water. So you can do this by posting placards, posting signs around the lake in the hope that it keeps people out of the water. And in most cases, it does. People don't want to go swimming in contaminated water bodies where they're potentially going to be coming out of those water bodies and itching for days to weeks. So that's a positive. Engaging in this particular approach keeps people out of the water. And for the most part, it ends up being relatively cost effective. But there are certainly a number of negatives regarding this approach. And the first is you're still going to experience that lack of tourism, which means you're still going to experience a lack of tourism dollars when we think about local businesses. In addition, remember, our point in this life cycle is as this dead-end host. So the parasites are going into us, but they don't fully develop. So even if you keep human beings out of the water, if you've still got birds on these water bodies and you still got snails on these water bodies, the problem is going to persist year or week after week after week, year after year after year. So it's not even a short-term solution to eliminating swimmer's itch in these water bodies. So other options, and these have certainly been employed in the past, you can go after the snail in the life cycle, with the idea being that if you control snail numbers, you potentially control the swimmer's itch parasites cycling through these really important intermediate hosts in the life cycle. And there have been a number of strategies that have been used to do this. The first, and this was actually used fairly extensively decades ago, it's not used so much now for reasons that I'll explain in a few minutes, but you can actually go in and utilize pesticides to eliminate snails in a given habitat. So copper-based compounds, copper sulfate-based compounds are really good at eliminating snails. And as I mentioned before, if you can get rid of snails in these habitats, you can potentially get rid of the parasites as well. So that's one potential option. Another potential option from the snail standpoint is to go in and modify their habitats. As most of you know, a lot of the key snails that are involved in the life cycle of avian schistosomes love macrophytes. They do extremely well on aquatic vegetation. So if you can go into these areas, reduce macrophyte numbers via harvesting, this can be mechanical harvesting via these large harvesters, or actually having people go out and harvest macrophytes. This can, in theory, reduce the number of snails in these areas, which in turn could reduce the probability of swimmer's itch actually coming in contact with human beings in these areas after the modification has taken place. So those are a couple of positives, and actually in the grand scheme of things, they end up being relatively simple, straightforward, and don't cost a lot of money to employ. So there are some benefits to employing these strategies. However, there are lots of negatives that go along with, in particular, pesticide application. Copper sulfate does a really good job of eliminating snails in these habitats, but it's also really good at killing off all sorts of non-target species as well. And so that's a big concern. And that's one of the reasons why copper sulfate cannot be used these days without some sort of a permit, because it has a general capacity to kill off all sorts of invertebrates in these areas, which generates lots of ecological risks. In addition, if you're utilizing pesticides, or if you're actually going in and harvesting in these areas, oftentimes this is going to be done on a relatively small spatial scale. And so even though you eliminate snails and macrophytes from a given area, it doesn't necessarily mean you've eliminated the problem, because if the wind's blowing from the wrong direction, from areas where snails are still occurring, you can actually have parasites, these parasite larval forms, being blown in from those areas as well, which in turn can infect individuals even in these areas that you think you've treated successfully. And then in addition to all of that, both of these methodologies end up being relatively temporary solutions. So just because you've added a pesticide this year and killed off all the snails, or you've harvested macrophytes this year, doesn't mean that those macrophytes aren't going to grow up next year, and next year the pesticides may not be having their effect. So you may end up actually having those snails recolonizing those habitats, which allows that life cycle to kick up and continue even in these treated areas. So you've got temporary issues associated with these kinds of methodologies. And then finally, the other possibility, the other target in this life cycle is the Mergansers themselves, which are key definitive hosts in maintaining these schistosomes throughout the Midwest. So there are a lot of strategies that have been employed here in Wisconsin as a way to reduce visitation of Mergansers on water bodies throughout this particular state and in adjacent states as well. Some of these strategies include trapping. So you can actually go out, you can set box traps, you can set funnel traps in the hopes that you can actually capture mergansers in their brood. And once you've done that, then you can actually move these mergansers to other habitats with the hopes that they stay in those habitats and don't return to the lakes that you end up wanting to swim in. In addition, a number of lake owners, lake managers have tried scare tactics, and this has included everything from lighting off fireworks to scare birds off of the lakes to firing off shotguns in the air to, again, scare these birds off of the lakes, hopefully into other habitats, and again, with the hopes that they don't return to their resident lakes. Now, some lake owners and lake managers have actually become even more desperate because of swimmer's itch outbreaks on their properties. And so they've employed more permanent sorts of approaches. So this includes actually walking along the periphery of lakes, finding common merganser nests, oftentimes found in trees, sometimes found in nests on the ground, and essentially destroying the eggs in these nests as a way to keep the numbers of birds down on these lakes. In addition, some lake owners and managers, I have heard, instead of firing shotguns up in the air, will actually turn the guns on the birds on the lakes and will actually end up killing adults and juveniles, again, as a way to try to control this problem. Because if the problem persists, oftentimes that means reduced revenues if they end up having cabins or hotels on the lakes. It also may mean lost dollar values in terms of the properties around these lakes as well. So people will oftentimes get desperate in terms of controlling the populations of hosts on these lakes. Now, there is another possibility that one can employ to try to reduce outbreaks of swimmer's itch, and that is to actually go about targeting the parasites. So as I mentioned before, adult parasites actually live in the blood vessels and the organs of these definitive hosts. And it turns out that there is a chemical that can be used successfully to eliminate the worms themselves within these hosts. And that chemical is Praziquantel. And Praziquantel has been used for decades as a way to control swimmer's itch, or not swimmer's itch, as a way to control human schistosomes in various areas around the globe. And it turns out that this chemical works really well against avian schistosomes too. So if you can get individuals out on these lakes and you can capture these common mergansers, and this has actually been shown via a number of papers that were published in the 80s and 90s, you can administer these birds with Praziquantel. You can do this orally, you can do this subcutaneously, and using the doses that they suggested, it ended up eliminating a very large number of the schistosomes that were residing within these particular birds. And so it turns out this approach has a lot of positives associated with it. So you can reduce the worms in these birds. And it turns out, based on the dosages that were being utilized, the hosts themselves do not appear to be adversely affected by Praziquantel, at least based on the dosages that they suggest. Another positive in this is that the drug is relatively cheap. So there isn't a large financial output in order to acquire this particular drug. And based on the work that was done in the 80s and the 90s, it was suggestive that Praziquantel might provide some level of a prolonged solution. So birds that were treated in one particular year, when they came back to their resident lake in subsequent years, they ended up having relatively low levels of infection. So there are a number of positives when we think about Praziquantel as a way to potentially control swimmer's itch issues. Now, that's not the perfect silver bullet by any stretch of the imagination. There's still costs associated with this. So you've still got to get people out on these lakes, capturing these birds and administering this chemical. But relative to some of the other approaches, this one seems like a pretty good option. And so what James and I decided to do was to develop a model around this mechanism of controlling swimmer's itch in the hopes that we could provide lake managers with a tool that would allow them to maximize their ability to control this problem and potentially minimize costs associated with trying to deal with swimmer's itch, both locally and across larger scales. So one of the things that we pride ourselves on when we're developing mathematical models is to make sure that our models do a good job of representing the biology. So I talked a little bit about the life cycle before. Remember, we've got these birds that are serving as definitive hosts. We've got snails that are serving as these intermediate hosts. And then we've got these parasite stages that are transmitting between those two major hosts in the life cycle. And so when we were developing this model conceptually, we wanted to set up a model that reflected these key features. And so here, as part of our model, we've got our bird population, and we can break our bird population into three major components. We've got our susceptible birds, we've got our infected birds, and then we've got this exposed category that represents birds that have recently become infected by schistosomes, but the schistosomes are not mature enough to begin releasing eggs into the aquatic environment. That occurs about 14 days after initial exposure, and at that point, birds are now transitioning into that I stage. We also want to represent our snail population. So here we've got our three snail categories as well: our susceptible snails, our exposed snails, and our infected snails. So again, we've got this intermediate stage that represents snails that have had that Myrissidia penetrate into them, but they're not yet releasing cercaria, and this ends up taking about 4 to 6 weeks. So once snails begin releasing those larval forms that go into birds and potentially go into us, they now transition into that I category. And then we've got our parasite stages. So the Myrissidial stage that's going to transition snails from that susceptible category into that exposed category. And on the other side of the life cycle, we've got beta that represents those cercaria that are being released from those infected snails that will now transition those susceptible birds into that exposed class. So our idea here was conceptually to set this model up in such a way that it did a nice job of representing the biology. And I think we actually did that quite well with the foundation of the model. So our big question was, how does Praziquantel, or the administration of Praziquantel, actually influence the dynamics of this system when you actually go in and treat birds within this particular system? So that's one particular aspect of developing these models. The second is to make sure that when you actually get to the point where you've got your parameters in place, that those parameters have some level of biological relevance. So how do you go about actually determining parameters for a model? Well, one of the first things you do is you actually recruit students. And this is something that we've done over the last 6 or 7 years. We've had a math biology REU at the University of Wisconsin-La Crosse. And as part of the projects that James and I have been involved in, we always try to get our students out into the field, collecting material like our cohort here. This is actually a cohort of our REU students down at Devil's Lake, right in the midst of one of those outbreaks. And here we are taking our chances that we're actually going to, potentially come down with swimmer's itch, collecting snails from a number of habitats around Devil's Lake. So once we ended up collecting those snails, we took those snails back to the laboratory. We determined infections in these snails, and those infection estimates provided us with a lot of really important information, both in terms of starting the model, so developing initial parameters for the model, and they were important in determining some of those transition parameters that I talked about a little bit earlier, determining levels of infection in snails early on, and also determining how the parasites might be dispersed from those snails to birds in the surrounding environment. So one of the other really nice things about getting students out into the field is that they oftentimes also provide you with very unique and astute perspectives. And this certainly happened with us when we were down at Devil's Lake. One of our students, Morgan, as we were sampling around the shoreline, noticed that there were no mergansers on Devil's Lake. And actually every time we went down to that lake to sample, we didn't see any evidence of mergansers. And so we talked to Morgan about that, and she started to investigate the migrational patterns of mergansers in a little more detail. So initially, when James and I were putting this model together, we envisioned this scenario where a lake like Devil's Lake serves as an established lake for these mergansers. They fly in the spring. That ends up being their resident lake through the summer and into the early fall. And then at the end of that period, they end up migrating away from Devil's Lake back down to their overwintering grounds. Well, Morgan, based on her observation and her digging, actually ended up finding out that, well, that wasn't quite right. That Devil's Lake was likely serving as a transient lake for these mergansers, where they would stop over for a few days or maybe a week, and then they would actually migrate to their resident lakes that were maybe a little bit farther north in Wisconsin, farther north in Minnesota, farther north in areas like Michigan and up into Canada. And so because of her observations, we actually ended up tweaking our model to incorporate this idea of transient lakes and resident lakes, along with incorporating some of the parameters these students actually ended up deriving from our field collections at Devil's Lake. And so now that I've set up kind of the biological background of the model, our conceptual model, and where we've gone in terms of conceptual development, I'm now going to turn things over to James, and he's going to talk to you about the nuances and the nitty-gritty of the model itself. So I'll turn it over to you, Jameser.
All right, thank you. Let me see if I can share my screen. I'll start where you left off, hopefully. Right there. Is that? And I do, let's see, view. All right, does that look okay?
That's great.
You hear me okay? Awesome. Well, so now I'll talk to you a little bit more about the model that we've created and some of the assumptions that went into it and some of the output that we generated from doing some simple simulations. So just as a reminder, you just saw that we're going to consider two different locations, a resident location and a transient location. The assumptions on the hosts and their presence at these locations will vary. And so let's start with the transient picture. At the stopover lake, we have the interaction possible between birds and snails happening. It's probably a little too cool for humans to be there having fun doing their recreational activities, but in that springtime, there is a possibility of parasite transmission. Now, what we're going to assume is in our model is that the birds are the ones that are bringing the parasite in, that the snails, if they had an infection going into the winter, that infection was, or that snail that had the infection is no longer around come spring. So the birds are bringing in a certain proportion of the birds are bringing in the parasite to the spring migration to both the transient lake and the resident lake. When they're there, they shed their eggs into the water. The snails pick them up, and by the time the snails start getting to that stage where they start releasing those cercaria, the birds have already gone on to their resident lake. So in terms of what's happening at the transient lake, it's a kind of a quiet system in the summertime because it's just now the dead end host, the humans, and our snails, until the birds come back in the fall, and now things can crank back up and get exciting again. So the important part of this is that for the transient lake, we're going to focus on just the snail part, snail host, because those are the ones that's the host that's going to kind of drive the model for at least the transient dynamic. So the transient model, like. So as I mentioned, the birds that are coming in, a certain proportion are infected either through overwintering at their overwintering site or picking up along the way as they go up towards their resident lake. No infection in the snail population, and the birds are gone by the time the snails are releasing the cercaria. The snails we assume that the birth rate reaches its peak in the late spring, early summer, and then tapers off throughout the summer and towards the fall. The interaction between the susceptible snail and infected bird, we assume that comes through the contact with the eggs, and that is a contact that just is only dependent on how many infected birds are coming through in the spring. And once the snails are exposed, they stay in that exposed category for about 5 to 7 weeks before they become infected and start releasing the cercaria. If we were to build this into a model, the common way we've done this with our students is to introduce a differential equations structure to the model, following a simple SIR type of structure where we have derivatives involving the important parts of the snail population. So we have the rate at which the susceptible snail population changes is based upon the births of the snails, and then the natural death, which is the mu, and then that transition from susceptible into exposed. So it'll reduce the snail population, increase the exposed population, and then we have a death rate due to a natural death rate for the exposed population, and then that transition from exposed into infected over a certain period of time. So each of these terms represent a transition from one category of infection to another category infection. And when we do some simulations to see what this looks like, we see some, I think, a pretty realistic picture. Before I talk about what the graph is, let me talk about the colors. So in some of the papers that were published talking about swimmer's itch and specifically the snail population that at some of these lakes, they identified prevalence of infected snails at these lakes with whether there's human cases of swimmer's itch. And they said that if you are above 2% of the snails in the lake or in a certain area with the parasite, then that usually corresponds with human cases of swimmer's itch. And so that's our red area here. If you're below 0.25%, then that's usually an ideal setting, and they don't seem to see any sort of correspondence with human cases of swimmer's itch. So that's the green, is that kind of the ideal range where no risk of having swimmer's itch cases at a lake. And then that yellow is the hit or miss. We don't know. It depends on the lake conditions. And so what we'd like to do when we talk about these simulations is keep, if we can, out of the red and ideally into the green. And we see that without any treatment, the infection levels or the proportion of snails that are infected rise in this first 30 days when the birds are migrating through the transient lake. And then once the birds are gone, the infection level dips down, starts to decay a bit. And then even though we start off with 0 snails infected in the spring, that's not the case in the fall. We have some that are infected when the birds come back, and we see a really large increase in the proportion of infected snails towards that fall migration, which gives us maybe concerns for later fall activities, but commonly human cases are quiet by that point. If we look at a resident model, now we have all 3 hosts in play. We have that parasite life cycle being able to complete itself. We have birds taking up residency for the summertime. We have the snails that are present, and we have the recreational period for humans. The birds, we assume when they arrive there, take about 21 days to lay their eggs, and before they go on and do other things. Those eggs take about a month or so to hatch and produce new susceptible birds. That's important for our model because they'll become a large increase in our number of susceptible birds. And then at the end of the fall, the birds will migrate away. We assume about 6 months from their arrival date. The way we describe the migration period and the increase in new susceptibles is by assuming that this migration period happens symmetrically, that we can assume that the number of birds that are arriving on a given day is roughly bell-shaped, with the peak arrival in the middle of that migration period. The number of new hatchings that come from that will depend on how many birds that have arrived at a given day, the sex ratio of those birds, and then the clutch size on that day. And then what will show up in our model later on in this as an increase in our susceptible population is those that survive to that adult stage, or at least the stage where they can become infected with this parasite. So we put this all into the model. We get a complicated system of equations, maybe not too complicated. Similar equations that we talked about earlier for the snail. The bird equations are representing or trying to reflect the fact that we have these changes from migration in and these parasite dynamics of susceptible birds becoming exposed birds, and exposed birds then becoming infected birds. So infected birds have 2 ways of increasing in value, either through migration during that first period of time in the spring or early summer, and then they could also increase when exposed birds start shedding the eggs, parasite eggs, into the water. So each term, I think, represents the biology and the form of the equations, or form of the model well. But there's a lot of parameters here. And as Greg says, that's an important part of our philosophy, is that we want to know where these parameters come from. Luckily, these birds and snails have been studied fairly well. And so life history data is out there to incorporate into the model, but transition data, knowing what the likelihood of an infected bird causing a new infection in a susceptible snail is not really well known. It's something we could try to tease apart in a laboratory setting, but it's often very difficult. Coming up with initial conditions that represent really what's going on at a specific lake, also very difficult. The initial condition part we can actually do, as Greg mentioned, by getting students and ourselves out in the field and making these observations. The transition parameters are a little bit harder. And so what we did is we relied on existing data. So about 5 or 6 years ago, a group out in Michigan were recruited by some homeowners that were concerned about Sturm Ditch cases at their lake. And so they brought in this research group and had them study the proportion of snails that were infected and offer up solutions. In this case, they actually applied Praziquantel to see what would happen to control Sturm Ditch at their lake. What's great about this is they have data for before they applied the treatment and after they applied the treatment. So before they applied the treatment, they took measurements of snail prevalence in 4 different locations. 10 different locations at 4 different time points in that summer. And so from that, we can use a method called genetic algorithm method to best estimate those parameters that we can't either tease apart ourselves, easily tease apart ourselves. And so, and if you want to know more about genetic algorithm method and how it works, please talk to me afterwards or put it in the chat and I can address it after the talk. It's a great method. But it gives us parameter values that best fit those observational points. The result of this gives us dynamics in the bird that seems pretty realistic to what we expect. We see a increase in both populations as the birds migrate in. Based upon our the data we found, we have a larger proportion of birds that are infected coming in, but then that doesn't stay that way for forever because those hatchlings come out and we see an increase in our susceptible bird population. And then once that hatching period stops, we have disease dynamics from the rest of the summer. And so that's why we see a slow decline in the susceptible birds and a slow increase in our infected birds. What's really concerning is that reflecting the data that we used, our graph for the proportion of snails that are infected is in that red during the period where we expect humans to be having a recreational time at these lakes. And so we would definitely correspond that with a high chance of having human cases of Sturm Ditch, which is what was observed at Higgins Lake. So getting back to what Greg mentioned and what really is motivating our study here is how do we do something about this? What can we do to treat this? And so our idea was to see how Praziquantel can have an effect, but Praziquantel is a drug that needs to be administered. And so when should that drug be administered? I think it's a natural question to ask. And we thought, well, mathematically, we should be able to do some simulations and answer this. And so this gave us the idea of optimal control problem, where we tried to decide how many birds should be treated each day so that we try to minimize one of 2 things, or a combination of 2 things. That is the number of Sturm Ditch cases that we expect to see at that lake, which we are going to assume is connected to how many infected snails there are and the density of infected snails at the lake. And the other factor that we're going to try to control in this is our costs. So even though the drug may not cost a lot of money, it does cost time and effort and money for the managers out there to administer this drug. The weight we put on those 2 factors, so reducing the number of Sturm Ditch cases and reducing the costs, is something we allowed ourselves to play around with a bit. So this idea that if we really want to emphasize reducing Sturm Ditch cases for a single season, so cost wasn't really a factor. So that would be if A is large, then we could look at those possible situations and what the simulations suggest our treatment should be. If costs are a factor, which they probably are most likely, then A is going to be smaller, and we can look at how that changes the dynamics of this system. So we did that in our system, and specifically we chose 2 values for A that would represent what we could visualize in our snail prevalence. That is, we chose an A so that a low value of A, so that the prevalence of snail was always out of that red area, out of that area where we expect human cases. So there, our study suggests we treat 21 birds. If you treat 21 birds in, this is the dotted graph, in those first 30 days or so, 40 days, then you have the greatest effect of reducing the number of swimmer's itch cases throughout the entire season. If you were to treat a little bit more, if you had more resources and money to apply more treatment, then you could actually bring the prevalence of snails all the way into the green area while humans were present at that lake. And that would be about 40 birds. This would be the dash curve for that. And about that same period of time when they're first arriving and are starting to lay their eggs and before the hatching and the burst in susceptibles come in. In both cases, we see a dip in the infected populations of birds represented by the bottom left graph. And so that's encouraging, right? So we could possibly know that if we could treat a reasonable number of birds, we could see a reduction of infected snails in the lake, and therefore less likelihood of having human cases. It's still a lot of birds though. And so an idea that we had, and this is again coming from our conversations with our students that were working with us at the time, is what would happen if we were to treat at the resident or at the transient lake, at this lake that the birds are hitting as they come through in the spring. And we saw that if you treat 15 birds at the transient lake, you're able to keep the number of infected snails, the proportion of infected snails at that lake in that green area while humans are present. So it's good for the transient lake. And the effect on the resident lake would be that this is the dash curve, would be that we wouldn't be below the red, which is good, above the green, so not ideal, but reasonable. So that by treating less birds earlier on at the transient lake, you could have a desired effect of at least reducing the likelihood of having swimmer's itch cases in humans. But becomes a problem with this. That is, by treating them early, even though there's fewer birds, by the end of the resident period, the number of birds that are infected are going to be higher than if you were to treat them at the resident lake. And so maybe that's not a concern. So the connection is with the snails that are shedding these parasites into the water. And so having a larger number of infected birds at the resident lake towards the end of the summer, maybe that's not as worrisome, but maybe worrisome for the future years. So if the birds are leaving that resident lake with a higher proportion of them with the parasite, they may return then in the spring with a higher proportion as well. And so even though you've treated the problem for that one season, we may see that a persistence of the parasite in future seasons. And so that becomes an issue too that we investigated, as well as how long should we wait to treat again once we treat in a single year? How long will that treatment last theoretically in our bird population? So as a quick little summary before I give and maybe some ideas for where we take this next, is that swimmer's itch is an emerging disease and days matter. So what our simulations seem to suggest is that we should treat these birds as soon as they start arriving. If you wait a week or 2 weeks after they have arrived to a resident lake, that is maybe what you can do, but that wouldn't be the ideal situation based upon our simulations. Treating early is good. So if you treat them when they come through on a transient lake, you can treat fewer of them, get away with treating fewer birds, but it may have an effect on future years because a larger proportion of birds are leaving with infection, which may mean that next year a larger will come back. And certainly any of the data on the systems we're studying is appreciated, and we should probably encourage more of it out there. What's nice is when we started this about 5 or 6 years ago, there was just a handful of sites that were collecting and publishing, showing their data. Now, when we revisited this with our students a year or two ago, there's a lot more of these lakes, which is kind of a mixed blessing. It means that it's still a concern, and it's still an issue that people are trying to handle. but at least they're sharing more data, and the more data that we have, the better our estimations and simulations can be. So before I end, I want to say two things. One, thank our students that were out there helping us and having these great ideas. And it's always fun to share our interest and love of ecology and these types of problems with them. The paper that has some more details in it is referenced below if you want to look at that in the future. And if you have to know any students that like math and like ecology and want to do these projects that are in the interface of these two areas, please send them our way. We are hosting the REU this upcoming summer in mathematical ecology. And so we're happy to have any students that you might think would be a good fit that would enjoy playing around with these problems on the river. So with that, I will stop sharing my screen and see if there's questions. I don't know how to stop sharing my screen though.
I can help you with that.
Okay, perfect.
Okay, excellent. Well, thank you very, very much for that, for your presentation and for sharing this with us. So now, you know, I neglected to mention it at the beginning of this, but if you to the audience now, if anybody has any questions, please put them in the chat. Greg and James will answer them, and so I'll kind of start reading them off. And those of you, and yeah, so please, if you have questions, please ask them. So first, we did see, okay, that was me. So there's kind of a question just about like kind of, do you want me to read them off, or I'll just read them off, like kind of about kind of some wildlife or diseases in general. It says, or I'm sorry, lake waterborne diseases in general, but it says several years ago, I heard about a young child that was swimming in either a large pond or a lake in northern Minnesota and inhaled through the nose water that contained a particular amoeba that remained in the headwater or in the head, later moving up into the brain and causing death. That there was little medical help that could halt the inevitable. Had not heard of this before, thinking the cold winters protected us in the north. Is this a rarity, or is this something to be concerned about?
So that is a unicellular eukaryotic parasite by the name of Naegleria fowleri. It is an amoeba, and it actually occurs in pretty much all water bodies across North America. But oftentimes they're found at relatively low densities. So it's only when the water temperatures get to the point where you end up having large aggregates of these amoebas where the potential rises for people to actually acquire them. They can swim through these areas, and that's absolutely true. If you end up getting this amoeboid up into your nasal passages, that provides them with the opportunity to actually migrate into and eventually destroy the brain. So it happens. There was actually an incident that happened about 5 months ago, a relatively recent incident in the Midwest. So it happens periodically, but it is very rare because you need essentially all of the planets to align in order for infections to actually take place in human beings.
Yeah, thumbs up. Awesome.
Thank you. Yeah, thank you. So next, and actually this was a question I was going to ask. I'm glad somebody else asked it. Is the common merganser the only host, or are other mergansers or waterfowl hosts too? And I'm going to follow up on that. Like if it's only the common merganser as opposed to like hooded or red-breasted, like what is it about the common merganser?
So a number of mergansers serve as hosts, and actually a number of waterfowl species across the board can serve as hosts. And oftentimes when you're talking about avian schistosome species, so Trichobelhartia is one that has the capacity to go into a lot of different merganser species. It just so happens that in a lot of the lakes and water bodies that we've been studying and looking at, common mergansers tend to dominate species-wise in those environments. When you get into other waterfowl, mallards, I think 60 to 70% of mallards have been shown to carry schistosome infections. Oftentimes they're different species of Trichobelhartia, and there are a number of other genera that can go through waterfowl as well. So yes, there are a number of different parasite avian schistosomes, a number of different species, and also lots of different waterfowl that can carry them. It just so happens that based on the work that's been done, the common merganser tends to be common in these areas, and at least based on the individuals that have been sampled, you can have prevalence values upwards of 70 to 80% in common mergansers.
Interesting. I don't have a, I just actually have a follow-up question to that then as well. So what is, and I apologize if you addressed this in your talk and I just missed it, but what is there any detrimental effect to the bird itself from carrying this parasite, or that we're aware of?
There can be. Yep. So if intensities get to be relatively high, because they actually live in the blood vessels, there can be problems. One of the issues with where these schistosomes live actually has to do with their reproductive biology. So females release eggs. If they're in close association with the intestinal tract, those eggs can eventually end up in the lumen and be shed out of the waterfowl. But a lot of those eggs will also circulate throughout the circulatory system. And there's the potential then for those eggs to actually lodge in major organs. And when that happens, the immune system of the host will actually target those eggs, which can lead to organ failure and eventually death. But oftentimes, you know, birds don't exhibit that level of mortality until intensities get pretty high. So hundreds of schistosomes within an infected bird. So yes, there are consequences. There are consequences, but I don't think that intensities get that high that often, to our knowledge.
Okay. So another question came in asking, is the U.S. Fish and Wildlife Service doing anything about people shooting the birds and destroying the nests? Or are you talking, like, were you talking, or I mean, I'm adding on to their question here, but is this an issue of private people taking matters into their own hands, or do you have managers that are actively controlling the birds through legal means?
It is primarily private lake owners or owners that have property around these lakes that I've heard have actually gone to these more extreme lengths to control these birds. I've certainly talked to my collaborators in Fish and Wildlife about it, and they've been certainly concerned. But that's about where it's ended. I don't think that they, because I personally do not know the specific lake owners or the lakes where this is being employed, so I haven't been able to direct them to particular individuals. This is all coming in through secondhand information.
Again, I just while we're waiting for other questions to come in, I have another of my own. Have you, so since, I forget exactly when you said this, you published your results on this, but have you found that there are like managers that are reaching out to you to like trying to adopt this model, like in elsewhere or besides Wisconsin? Or is it, have you seen places where they're actually putting this into practice a little bit more?
Do you want to take that one, James?
Sure, I can try. So this book got published in February-ish, sometime early this year. So the short answer is no. The long answer is that we hope that it will be soon. That in the past, a lot of times when they were going out and putting out this treatment of Praziquantel, it was just convenience, based on convenience and not based on any sort of mathematical tool that they had in front of them. And so the hope would be that we could address it in that way, that we have given them some tool to do. And just as related to this, in our paper, we also discuss what happens if you don't have the ability to go out every single day and apply this treatment. So as you saw in what we presented, this was in the idealized situation where we have a continuous treatment regime, and that's probably not feasible for our wildlife managers. And so we did another study where we talked about if you can only treat on 2 days, when should those 2 days be in when the birds arrive to have the greatest effect? So our hope, our intention is to have some practical results and conclusions that can be used, but we haven't really heard much about them being used just yet.
From a scientific standpoint, I think it's garnered quite a bit of interest.
Yes.
From other modelers.
Yeah, that's true. We've gotten some positive feedback from some of the people that we were referring to in our paper, and the people that have studied these birds on the Michigan lakes.
That's it's fascinating. I mean, I would think that, you know, if you can find a way to resolve this issue with this nasty, disease, you'd certainly want to do something about it. There's one other question. I think this is a good one. It says, are there any known positive aspects to the existence of flatworms in the current ecosystem?
Oh, that's a question that's near and dear to my heart. That's a great question. Probably. Yeah, probably. I mean, even the fact that you may end up having these flatworms impacting certain host genotypes, whether it be at the Merganser level or at the snail levels, you could have the parasite actually aiding in the control of population levels of these hosts. So, and not just that, I mean, you could also have these parasites potentially outcompeting other parasite species that might actually have greater adverse effects on these ecosystems. Is that actually known? No, but certainly parasites are known to interact with a lot of different species, both in terms of other parasites and other hosts. And removing certain parasites from systems can actually have detrimental consequences. And that's something that we've gone back and forth with as well. So what happens if we actually do eliminate schistosomes from these habitats? Is it going to have a major impact on these ecosystems? My feeling is, given how few hosts are actually really adversely impacted by these parasites, at least at the definitive host level, and how few snails in the grand scheme of things are actually infected by these parasites, that the elimination of schistosomes from these habitats probably wouldn't lead to the unraveling of the ecosystem, but it could have more nuanced impacts if you eliminated them completely. Yeah.
It's kind of like the question of like, should we eliminate mosquitoes? Like, is there,
right.
It's like, is that an ecologically safe thing to do, even though they spread disease and are pretty annoying to us? And
absolutely. And actually the other aspect too that is a concern is that when these snails are infected, and as you saw by those pictures, the number of parasites that are shed from those snails, we're talking about thousands, tens of thousands, hundreds of thousands. And a lot of those cercaria actually serve as food for aquatic organisms, invertebrates, and a lot of fish species. So, I mean, there could be impacts at that level too, if you eliminate those schistosomes.
that was actually going to be my next question. Is there anything that you know of that feeds directly on these schistosomes or on the flatworms themselves, or anything, if there's something that depends on them for food?
You bet. So there are actually a number of invertebrates that rely extensively on those larval worms as part of their diets.
Okay. Well, I'm going to go ahead and keep this open still for a few more minutes. If we're only a little bit after 5, so if you guys are welcome to stick around. We have, if for the students in the group, if you have class and so forth to get to, feel free.